Welcome to an Achieving Science video. This video is going to cover genetic engineering. So first of all, what is genetic engineering? Well, genetic engineering is used to change an organism and give it new characteristics which we want to see. So in order to do this, we need to look at DNA. So therefore, it involves changing the genetic material, which is referred to as the genome of an organism. Now what can happen is genes can be transferred into both animals and into plants. Now why do this? Well, first of all, let's look at some advantages of doing this. Well, one advantage is that bacteria can make the exact proteins needed, in the amounts needed, and in batches that are very, very pure. Fungus cells can be used to produce the hormone insulin, which is needed for people with diabetes. It can improve the growth rates or yields of plants and animals. Therefore, this adds values to crops and reduces the amount of fat in animals. We can do it so plants are able to produce their own pesticides, so we don't need to spray plants. Food lasts longer in supermarkets. Plants can be designed to grow in hot, dry conditions, and so help with problems like world hunger. But of course, with anything, there are also some disadvantages. So let's now have a look at what these disadvantages include. Of course, this is a very new science, so we don't actually know what the future holds and what the impacts could be. Therefore, there is unknown effects on human health. With plants, we could have the spreading of GM genes, genetically modified genes, with those of non-genetically modified crops. There is the danger that we could actually manipulate genes to ensure a child is born healthy with good sporting ability. There is also concern over the effects of wild flowers and insects. There's also concern over the possible effects on human health. So what about the future? Well, in the future, it's hoped that it's a positive experience and a positively used technique. So it's hoped that people with genetic diseases can be treated by injecting them with the correct genes, i.e. for genetic disorders such as cystic fibrosis.